Executive Council webinar, Transitioning New Safety Biomarkers into Routine IND-Enabling Studies in Early Development. Today's session is scheduled to go 60 minutes. I am joined by my esteemed colleague, Dr. Jack Reynolds, who will be moderating yet another great roster of panelists who, by the way, should be dialing in right now with their special panelist passcode. I will introduce all of them in just a moment. If you are listening through your computer, which I hope most of you are, and you're having any kind of audio problems, meaning you can't hear our voices well, we do have a dial-in number as a backup, and that's on the screen. That's on the screen in front of you. Our hope is to have today's session recorded, and if technology cooperates, we'll be able to place this on drugsafetycouncil.org shortly following today's session. Also, a couple of other housekeeping announcements. We will be taking questions from all of you. We have obviously a large audience today, so you will be in listen-only mode except for our panelists, but we ask you, you could certainly ask the panelists questions through our Q&A module at the top of the screen. I will attempt to point towards it, and you could type in a single line field of your question. We'll put it into the queue, and we will answer it in the order that's received. We allot usually 10 to 15 minutes at the end of our session to take questions from all of you. At that time, we'll also put up a feedback form that we hope that you could complete. It gives us guidance on what we can do better, and equally as important, what kind of topics we could cover as the Drug Safety Executive Council moves forward into the year with our monthly webinars. So it's a great, useful way to gather your feedback, and enables us to create more valuable sessions for all of you. If you are new to us, in short, DSEC, or the Drug Safety Executive Council, is a free online community. It is limited to representatives of biopharmaceutical companies. However, from time to time, we do hold webinars and open it up to guests in the life science community that represent regulatory agencies, academia, technology providers, and so forth. And today is one of those sessions. Just a little bit of background on who DSEC is. DSEC is a community owned and operated by Cambridge Health Tech Associates. We are a unique consulting company. We focus primarily on bringing folks together in life sciences, specifically through consortia and communities. We help pharma work together to explore new technologies and processes, reducing costs and improving overall due diligence approaches. We also work with technology providers in bringing them in front of pharmaceutical companies, increasing their access to market, hopefully their access to their ability to market and sell to groups of pharmaceutical companies by bringing them in partnership to form groups of pharmaceutical experts. Our two communities, the Drug Safety Executive Council and our latest community in the genomics space called NGS Leaders, both are free to join, and in fact NGS Leaders is open to not only biopharma companies but academia, technology providers, and there's actually no affiliate restrictions on how to join there. And both communities are designed similarly to facilitate peer-to-peer consulting, promote thought leadership, and also promote relationship building and lead generation. CHA also boasts 3 industry consortia. We have an ongoing consortium around the evaluation of new technologies. We are actually in the process of launching our second consortium around the accessing of safety data in the GLP space, and we are actually working now on a pre-launch of a third consortium that would help validate software that's being used in GLP studies. As it relates to today's topic, I wanted to give some background on our Technology Evaluation Consortium and put some context into that. Our focus is on identifying, validating, and then adopting novel predictive safety technologies, and we've put together a group of 11 biopharmaceutical companies who have helped contribute to a shared library of compounds, all of which have correlated in vivo safety data attached to it. And our objective is to partner with technology providers that have unique novel platforms and work with them to determine if any modifications should be made to that platform, assess the scientific utility, and then, of course, the objective, obviously, is for pharmaceutical companies if they believe there is a high level of benefits to the platform that they get into individual commercial arrangements with the providers. It dramatically reduces the costs and time that pharma puts into the due diligence of these technologies. It dramatically reduces their risk, but it also helps obviously expedite the commercial adoption of these new technologies from the technology provider's perspective. So we like to call it a win-win type of arrangement, and that consortium is in its second year and ongoing. At a high level, we are owned and operated by CHI, the conference company, who you guys know is a leader in conferences and reports, and that's really where we sit. I want to introduce to you, prior to starting here, a colleague of mine on the DSEC team to also give you a quick little overview of a new complementary program that we've recently launched that may be relevant to many of you, especially those of you new to DSEC. So I'm going to introduce to you our Head of Business Development and Strategic Partnerships, Brian Murphy. Brian, are you there?
I'm here, Eric.
Go ahead.
Great. Well, so in the spirit of helping bridge the gap between drug developers and technology providers, DSEC recently launched a virtual poster gallery, and this is a forum for biopharma companies to learn more about innovative tools and techniques in drug safety to make your job easier. And it's also a medium for the vendor community to build awareness of emerging technologies and methods. Excuse me. The gallery is free, both to view and to upload a poster. And to view the site, simply go to our website, which again is drugsafetycouncil.org, and click the tab for the poster program. Several companies have already contributed posters, and we're getting new ones each week, so we encourage you to visit that frequently. So if you are developing a new technology or method relevant to drug safety, simply go to the site to upload your poster as a PDF. The poster is first reviewed by DSEC before being displayed in the gallery, and it will remain on the site as long as it remains current to the community. Also, I'm very pleased to report today that we'll soon be announcing 4 nominees for DSEC's Technology Innovation Award for the first quarter of 2011. And nominees are chosen from the poster gallery and are also determined based on the merits of the science and its application to the field of drug safety. Nominees will be announced to the candidates and also to the DSEC members next week, and the overall winner will be selected via a vote from the DSEC members. So for all of you out there, I encourage you to go ahead and upload your poster as soon as you can. So I think that's it, but certainly feel free to contact Eric or myself directly if you have any questions on the gallery.
Yeah, thanks, Brian. I appreciate it. And so DSEC members, be on the lookout. We'll be sending an email next week around our 4 nominees, and we'll be putting together a 1-question poll that you could vote on who you think the winner should be, as Brian said. And for those of you new to us, especially those in the technology space, feel free to go to our homepage, drugsafetycouncil.org, click on the poster gallery button, and feel free to upload for no charge. Of course, there's no charge, there's no hook, there's no credit cards, you need nothing like that, and you can upload your poster in PDF format right up to the website. We'll take a look at it and by and large usually approve it and get it right up there for our members to review. So with all that, I'm going to forewarn our panelists that I'm going to do some introductions right now, so you may get ready to unmute your phones. You guys have been great keeping them on mute. I could hear there's very little background noise, so that's terrific. I appreciate it. And I'm going to start with the introductions. Jack, I will save you for last. And I guess in alphabetical order, I'll introduce our esteemed panel, and we'll get right into the session. A reminder, everyone, and I'll probably chime in a couple of times throughout the hour, ask questions. We'd love to get questions from all of you. And also, please be on the lookout for our feedback form at the end of today's session. So our first panelist representing the FDA is Dr. Daryl Abernethy. He is responsible for leading the development of pharmacology mechanism-based safety programs in the Office of Clinical Pharmacology. He works in synergy with efforts in the Office of Safety Surveillance and Epidemiology and other offices and centers around the FDA. He brings more than 25 years of experience in medicine and pharmacology, including positions in academia, practice, and research. And prior to joining the agency, he served as Chief Science Officer at USP. Daryl, thank you so much for joining us again. We really appreciate it. Have you been able to dial in okay?
Yes, I'm hearing you, and I'm glad to be here.
Great. Thanks so much. Our second panelist is our colleague here at the Drug Safety Executive Council, Dr. Ernie Bush. He runs all of our scientific programs and collaborative projects. Prior to convincing him to join us, he had spent over 23 years working in biomedical R&D. From 92 to 2005, he was at Hoffman LaRoche. During that time, the last 5 years, he led their non-clinical safety department in Nutley. So, Ernie, thank you for joining us. I'm assuming you dialed in safely?
Yes. Good morning, good afternoon, everyone.
Our next panelist is Dr. Elizabeth Gribble-Walker. She joined the Critical Path Institute as the Assistant Director of Predictive Safety, the Predictive Safety Testing Consortium in January 2008. Her professional experience includes preclinical program development and oversight of biologic drugs for autoimmune and oncology indications, design and interpretation of mechanistic toxicology, combination safety, and GLP toxicology studies, and medical writing in support of regulatory submissions. We're very pleased to have been able to fit on her calendar. Thank you, Elizabeth, for joining us. Have you been able to dial in?
I'm here. Thanks so much for the invitation.
Thanks. Last and certainly not least in representing industry is Dr. Tanya Zapta. She is the Global Safety Biomarker Lead and Senior Veterinarian Pathologist at Roche. She is, of course, a board-certified anatomic pathologist, training down at the University of Georgia Veterinary Medical College and University of California, Davis. She joined Roche in 2007 as a veterinary pathologist in non-clinical safety, becoming the lead of global safety biomarker initiatives in the spring of 2010. Tanya, thank you for joining us.
Thank you. I'm here.
Great. And finally, our moderator, chairman of our advisory board, most of you know him, Dr. Jack Reynolds, the former global head of safety at Pfizer, and who's been helping us with the Drug Safety Executive Council and leading our programming for, now going on 4 years. So, Jack, I will hand it off to you. I'll put up your slide here, and you can take it away.
Okay. Thanks so much, Eric, and thanks very much to the panelists for taking time from their busy schedules to join us. I'm going to go very quickly through this slide, but I just wanted to set the context for today's discussion as participants on the line have broad and varied backgrounds. So really the question we're asking today is how can we leverage our new knowledge of disease or adverse event biology to enable improved clinical trial design? When we refer to biomarkers specifically, and the topic for today is broader than biomarkers, but we have to realize that these are decision-making tools, and many of them have existed for decades. So I think we have a number of precedents in place for some of the new and emerging biomarkers. What we refer to broadly in FDA's new guidance document refers to drug development tools. For some of us that have been around for a while, the moniker DDT has a certain ominous ring to it, but I think it really does connote very well what we're talking about. If you get down to the very basic level of what we're trying to do, we're really trying to measure, and in terms of the translation of these drug development tools, we're trying to measure biological responses across multiple scales, and beginning at the molecular level in vitro. We want to look at these markers at the cell level as well, organ or whole animal to the human. So the difficulty in many cases is to translate or to carry these measurements across these different scaled processes. I like to think of these as surrogate measures for a biological response. Certainly in my career, used to be when someone would mention a biomarker, it was the kind of thing that needed to be formalized, create a department around it, and create a head of biomarkers. It really is an elementary process looking for a measurement of a biological response. The process, of course, is characterization of that response at the systems level, if you will. What do we want to measure along a response pathway? We need to validate that measurement. We need to validate the technology or the methods. Clearly, this validation is separate from validation of devices or instruments that would measure these, so this really is the method. And importantly, and I think the essence of what we want to discuss today is really the qualification of these biological response indicators for decision-making. They are distinguished around efficacy biomarkers or efficacy measurements or safety measurements. I think for many of us, a big breakthrough in the process of identifying, validating, and qualifying biomarkers has been the FDA's promulgation of what they refer to as the context of use or fit for purpose. Many people have felt that this response measurement needed to be validated all the way through to humans, et cetera, before it could actually be of utility. And certainly FDA has really promulgated, and in their new guidance documents clearly states this can be a staged process starting with the most elementary form of the measurement. Many of you certainly identify that having these drug development tools really serve as a foundation for the essence of personalized medicine, and certainly for us in the Drug Safety Executive Council, these drug development tools provide opportunities for personalized safety as well. So with that, trying to get everybody oriented, I'll quickly go into the questions, and we intend for the panel discussion to be a discussion or a dialogue. So please, with the panelists, I will start by referring specific questions to each of the panelists, but please, panelists, feel free to come back with a follow-up response or a further discussion of what the question is. So, and again, I would invite our participants to use the upper left Q&A drop-down menu to submit questions to us, and we'll try to get as many of those as we can through this session as well. So very quickly going into the questions then, the first one is, what new preclinical safety biomarkers are you using routinely or exploring for routine use? And obviously we have a broad area of where people are working, so please, from your perspective, whether it's the FDA or Critical Path Institute, please give us that. So the first question in, Tanya, maybe we'll have you respond. What new preclinical safety biomarkers are you using or exploring in your organization?
So probably like most pharma, there's kind of two approaches. Maybe there's we try and develop a toolbox to have kind of an organ-based approach. So likely organs that are common targets for the type of drugs you're developing. And it also then allows for a more project-based, quicker response if you have that toolbox existing, which is more the difficult arm for that. So from a project-based response, usually if you don't have a biomarker that's qualified and validated, so for instance now with the kidney for proximal tubular injury, you're either looking to back translate a marker that's available in humans and see if you can use it quickly in animals. So for instance, you might use thyroid hormones or bone biomarkers that are more commonly used in humans and back translate them to other species like rat or monkey. The other approach would also be to use a biomarker that's already qualified and validated for another use, because it's easier to probably bridge that and show the scientific rationale and support for putting it into a safety study. So for that instance, we've used gastrin for chief cell degeneration. So you could do it in that respect. And then there's the organ-based approaches, which is what the PSTC and the IMI Safety Consortia are doing, as well as ILSI-HESI. So then you're just trying to look at what are markers that are specific and or sensitive for the heart, liver, kidney, skeletal muscles. So I say, we participate in those consortia, and so a lot of our work is to support those as well. Okay, thank you very much. Liz, certainly the Predictive Safety Testing Consortium and the Critical Path Institute has been active in this area. What biomarkers are you working on or exploring in your consortium?
Certainly, and Tanya gave a great overview, as she said, of not just PSTC, but some other organizations with similar interests. So PSTC has a number of working groups that are formed around the priority interest and safety biomarker needs from our membership, which is currently 17 pharmaceutical companies. Those areas are, I'll just repeat what Tanya said, they're kidney, liver, skeletal muscle, cardiac hypertrophy, and then vascular injury as well. And among those different target organ areas, our process for nomination of a safety candidate biomarker for evaluation under the consortium framework is really that at least one, if not several, of our participating member companies have already generated a fair amount of internal experience, both with a functional, technically valid assay, as well as with an understanding of sort of the biological response of the biomarker to answer the safety question of interest. So what I can say is among those 5 different working areas that we have currently, there are currently another 21 biomarkers of kidney injury that are being evaluated. For liver, we have 6 biomarkers that are formally being evaluated currently. For skeletal muscle, I believe there's another 5, and for cardiac hypertrophy, they're focusing on 1, which is NT-proANP, as an analogous biomarker to how BNP is currently used in the clinic. So rather than perhaps going through all of those biomarkers and listing them, most of those are available publicly on CPAP's website, and I would just direct the audience to kind of follow up there to see what exactly it is that we're looking at. What I will note is, I said that the preferred method for how we go about evaluating a biomarker is sort of this low-hanging fruit or opportunistic approach, where there may already be a fair amount of data out in the literature, and again, the internal experience at our member companies as to the utility of this biomarker. But it's really, and again, this is the power of the consortium approach, it's when that biomarker can be brought forward, that information shared, and ultimately a very large data set of potentially something like 20 to 40 model compounds, both sort of known prototypical toxic agents and proprietary compounds from our companies, that those tox studies can be run and data all compiled together, really, I think, allows us the confidence to understand the true utility of a biomarker and its behavior in terms of kinetics, how it develops with progression of injury, injury reversibility, et cetera.
Okay, Liz, thanks very much. So, Daryl, many of our participants online probably are not aware of your initiative that you came on board FDA to establish. So, please, if you wouldn't mind, give us a brief update on that, and maybe you could share with us some of the things that you're working on in the near term.
Very brief. For about the last year and a half, Larry Lesko and I have been working on a new program here at FDA that has to do with mechanism-based safety prediction, bridging preclinical and clinical areas, and bridging pre-marketing and post-marketing. We've generated a good amount of interest across academia, the pharmaceutical industry, and from about 10 companies at this point, and NIH to move this forward. It's an early effort to move safety science as it translates into the marketing and into the pre-marketing and post-marketing clinical arena, from what's really a pharmacoepidemiologic pharmacovigilance approach to using mechanism-based safety to inform that activity and to create hypotheses that will assist the epidemiologist and also to predict. To that end, I guess about 2 weeks ago, during one of our snowstorms here, we had a meeting on a couple weeks ago, Thursday and Friday, and it wasn't a truly open meeting. About 80 people who we'd identified who were working directly in this kind of an area. from a number of your companies, for those people who are on the line, a number of academic centers who have leading programs in bioinformatics and cheminformatics and systems biology, and then a number of the institutes at NIH were engaged. We're moving that forward and developing now specific projects, what the people at the meeting like to call use cases, to evolve the concepts further in specific areas. And at the moment, we're identifying 2 or 3 specific areas, and then the companies who have interest, the academics who have interest, and so on to work in those areas. We will be working with to coordinate and push it forward.
Excellent, Gerald. Thanks very much for sharing that with us. Ernie, you have seen a lot in your efforts on leading collaborative projects and your scientific activities. What do you see as those biomarkers or drug development tools that are being routinely used or explored?
Yes, Jack, I think most everybody on the line is probably familiar that we do a lot of polling and survey questions as part of the DSEC community to try to keep a finger on the pulse of new technologies and new activities. And for those of you, we just sent out our annual new technology survey, and if you haven't filled it out, we would like to encourage you to do that and send it back. And one of the points, so it's decidedly on a survey level or on a broad industry level, it's a decidedly mixed message with regards to biomarkers. It's consistently over the past several years, matter of fact, I think literally since we began surveying the new technology trends, biomarkers have consistently been one of the top areas of interest expressed by the responders of the survey. Often the top area or within the top 3, and particularly, of course, the liver or hepatotox biomarkers is consistently rated as one of those areas where people really would like to see further development or new developments. So the general level of interest and support for biomarkers remains extremely high, although we haven't looked at the 2011 numbers yet, at least through last year. They were very high, and that's the good news, I think. But also on a level where we ask more detailed questions about what are you actually, what is the new biomarkers you've actually implemented, trying to get a feel for those compounds that have been nominated, for instance, through the predictive safety testing consortium. Is there beginning to see an implementation of those in routine or semi-routine tox studies? And indeed, we are seeing some people pick some of those up, but I have to say the counterweight, it's not really bad news, but maybe slightly disappointing news is by far the preponderance of people respond when you ask them what's the most recent biomarker you've implemented in a routine or semi-routine way for your safety testing is troponin for cardiovascular safety or different troponin subclasses. So I have to say, even though it's been 5 years since the priority list, or getting very close to 5 years in March when the original priority list for the Critical Path Initiative came out and biomarkers were definitely highlighted in that list, we're still not seeing a rapid uptake of biomarkers into routine or semi-routine use. And what part of the discussion would be nice today is to see if there's ways maybe we can help bring those forward even faster. That's it, Jack.
Okay. Thanks, Ernie. I do think there has been a lag time, if you will, in biomarkers, but I certainly think activities on the part of FDA and IMI, as well as several consortia, I think are going to kick up the speed of this process considerably. So going to the second question, is it essential that preclinical safety biomarkers have both preclinical and clinical validation and qualification before you would consider using them in your preclinical safety studies? So I think the new FDA guidance document, as we have referred to, addresses some of this, but it would be nice to have a varying opinion from different sources. So, Daryl, I know you're in the middle of this, but perhaps you could share with us what your view is to the need for validation qualification before one begins to use a biomarker preclinically or clinically.
Well, first, I'll offer my opinion. There are probably many opinions within these walls. But clearly it would be unfortunate to not be using a variety of safety biomarkers that appear to have utility in the development phase, even when they had not had a huge experience clinically. At the same time, of course, the more that bridging can occur, the more effectively, or I guess you'd say, the more confidence that one has. But I think that a general sense, and it's kind of what you reflected on from that draft guidance, is that we want to encourage people to explore and to broaden the array of biomarkers that they're able to use, and the only way that will happen is really from the ground up. And so in the preclinical arena, to see what looks promising, to see how that translates into the clinical arena would be the longer-term goal.
Excellent. Thanks, Daryl. Tanya, what's your feeling about early validation qualification before you move forward with using a biomarker?
I think it's what we strive to have, because there's no reason to really validate something in the rat if you can't find a similar endpoint in humans. So you're using it to get into humans. So it's not very helpful if you can't find a similar marker in humans. Certainly, what we try to do is when we prioritize which biomarkers to select and to develop or bring in-house is to try and make sure they're translatable and they have utility in the clinic, and that just means having a stronger alignment with our clinical colleagues early on. And that's certainly not an easy task, but something that we're striving to do.
Okay.
But I would also say that the reverse translation is also important, so to get the feedback from the clinical data back into the preclinical setting is very important and not always easy as well. So if you've used a marker in your preclinical setting, it's nice to know how it worked in a clinical setting so that you know and you can learn from it and grow and do some progressive qualification and validation. So the forward and back translation, I think, is critical to having an effective biomarker strategy.
Yeah, I would just emphasize, Tanya, a great point you made. Since I think the Drug Safety Executive Council has tried to do is to bridge the clinical to preclinical and preclinical to clinical gap, but certainly feedback from the clinical groups have been extremely important to many folks, and I would just emphasize that's one of the things we try to do and would encourage folks to have these conversations with your clinical people or the clinical people to speak with the preclinical folks. So, Ernie, what's your feeling towards qualification of biomarkers?
Yeah, I would have to support from what I'd call sort of in practice level of what Tanya just said. The fact of the matter is the PSTC produced a very nice set, and for all practical purposes, the agency agreed that there were kidney biomarkers that were probably value added for preclinical studies, and yet our uptake data tends to suggest they haven't really been widely or even partially put into practice in most major pharmaceutical companies for routine preclinical tox studies. So I think the practice has told us rather definitively that it is essential that you have clinical validation before they're going to routinely put these biomarkers into their GLP tox studies.
And I think the difficulty about immediately even using that nice data from the PSTC is that, you know, it's clearly indicated that it's qualified for a certain type of injury in the kidney. And, you know, while a lot of compounds that is the endpoint, many compounds that it's not. And so, that's, I think, eventually where there's questions where we could get guidance from the regulatory bodies of how much will you hold the program to if you're trying a biomarker for an endpoint for which it's not qualified. And so if it doesn't corroborate the histology, or, basically is it going to have a negative impact? And that's the problem with introducing biomarkers, especially as a screening tool, because we don't actually know enough about how they behave outside of what they were specifically tested for. And some pharma know more than others through internal use, probably through their discovery studies or model studies. But gaining that experience is going to take a lot of years, and if we can't use them without penalty in our safety studies, then especially in large animals, where you have a very small N number, so you can't just go running, qualification studies on dogs and monkeys, that there has to be probably some discussion around how we can use them truly as an exploratory tool, and they do not follow that compound around. They were just purely used to understand how they would respond through studies.
Yeah, let me just say, I think that's a really nice idea, and the sort of, if we can refer to the toxicogenomics arena, the sort of safe harbor concept to encourage people to utilize these new biomarkers, but not in a way where they're going to pay a penalty if, in fact, you get surprising results.
So if I could also interject some comments.
Yeah, Lisa, I was going to go over you next.
Thanks. Sure. So I guess a number of just points I'd like to try to make quickly. So to answer the question as it was articulated, so is it essential that preclinical safety biomarkers have both preclinical and clinical validation before you would consider using them in preclinical safety studies? So just to recap for the audience, the 7 kidney biomarkers were originally qualified for use for detection of acute kidney injury in the rat, and as Tanya said, they were linked specifically to either glomerular or tubular or both degeneration and necrosis, so a very specific histopathological diagnosis. And I think Tanya's point about the ability to, if you have a slightly different finding with a new drug, the ability to perhaps use biomarkers as a starting point for exploring whether or not they appear to also sensitively monitor your slightly different and particular injury of interest. I think that's a very important path that needs to be on the table, because, as she said, the characterization, even with this first set of studies, 23 some odd rat tox studies, there's going to be an ongoing process of our familiarization and learning more and more about these biomarkers' behavior. However, I wanted to also note that in a survey of the PSTC membership, and this was 16 members at the time, we did see 100% of the companies state that they would use these biomarkers preclinically if an issue with nephrotoxicity arose, and 9 of those companies had already, unfortunately, had the opportunity to do so preclinically. What was very interesting was that 50% also indicated that they were already using these biomarkers in the clinic. So although the language that both FDA and EMA and PMDA reported back regarding the qualification was that these biomarkers were qualified for use preclinically and on a case-by-case basis in the clinic, 8 of our 16 companies were already using these on active programs in the clinic, and that was in response for about 2/3 of those. That was in response to the qualification that gave them the confidence to do that. The other ones were already using them clinically. So that may contrast a little bit, Ernie, with it sounds like I think you definitely have other entities among those who you surveyed about the use of these biomarkers. Another signal, I think, about their use is Federico Goodstead has recently reported as he searched through FDA's IND documents and reports. So again, these are FDA reports, not actual INDs coming in, but he was able to find 42 references of KIM-1. So we searched just specifically on KIM-1, which is generally understood to be the most kind of popular of this set of biomarkers. And that the timing of those mentions of the kidney biomarkers was such that there was a significant uptake in frequency of KIM-1 following the qualification itself. So I think that's a very positive signal for FDA and for all of industry and those of us vested in this qualification process that, yes, qualification does mean something in terms of the utility and uptake of these biomarkers. And then I just wanted to also state briefly, in the survey of the membership that I mentioned about use of the kidney biomarkers, you did see fairly, some large differences, I think, in terms of how they were being used. It was clear that some companies were indeed using these biomarkers in lead optimization or screening efforts, but that others were still sort of reserving them for monitoring purposes once a potential kidney liability had been identified via sort of more standard methods, i.e. histopathology.
Okay, Liz, thanks very much for that. We want to go to the third question, but Eric, you wanted to make some comments about some polling questions?
Yeah, I was just going to just forewarn our panelists. We'll put this question up, and then for our attendees, we're going to actually ask you a few questions. Make sure you're not necessarily sitting on your hands. There'll be interactive polling questions that we'll throw up from time to time throughout the last 10 minutes or so, and then that feedback form will come up at the very end as well.
Okay.
So
the third
actually,
question.
Anya, I just want to make one comment in response to what Liz said. Is I think interesting that your polls showed they were used in lead optimization or as second tier after you find you have a kidney liability. And I think that's what I was trying to mention, is that there's really not much in between, because at one point, can you use it as part of your standard panel to gain understanding of what the biomarker is doing? And I think, especially knowing that qualification can take many years, and it's not even qualified for multiple endpoints within an organ, that's, I think, determining how we can do that will allow us to move forward faster.
I would simply say that we're currently under discussion of what a safe harbor program might look like and what sort of internal changes would need to occur for the exploration of not only biomarkers but other sort of safety data coming from other sources. And that's just at the beginning process. But I guess what I'm trying to say is that I certainly hear loud and clear what you're saying. It makes a lot of sense, and something that we're working toward making easier.
That's great.
That's great to hear. I mean, a lot of the concern that we hear is that as we develop more sensitive biomarkers, it really potentially opens up this question about almost a philosophy about what is toxicity, what is truly physiologically relevant in terms of novel biomarkers that we know are much more sensitive than the ones we've used for years and years. So I think certainly that there's an extreme level of trust necessary for companies to begin to engage in dialogue with FDA and other regulatory bodies around this.
Yeah, I'd just like to add, I don't really see a conflict between the DSEC results and the PSTC results at all. The DSEC results specifically talked about routine usage, and the PSTC was specifically qualified as something like when there's a signal or when there's a need. And I think both of those numbers are, and the discussion afterward, both of those numbers are probably realistic snapshots of the current situation. That yes, when people need a more powerful tool, they're going to these new biomarkers, but as part of their regular GLP tox platform or range of compounds they routinely measure, they're probably not yet using these biomarkers to any significant degree.
And I would think, Ernie, as well, that cost certainly does play into that. Most of these biomarkers are still, well, the multiplex from RBM, panels from MSD are the most common platforms that we're evaluating, and again, compared to routine measures, the cost is somewhat potentially prohibitive for running them, as you said, on a routine basis.
That's interesting. Yeah, we didn't ask that question, but maybe that is something we could add. Thank you.
Okay, so the third question, and then Eric will put up the polling question while we're doing this, but is referring to the FDA's guidance document for the qualification process for drug development tools. So we'd just ask the folks to, from your perspective of your organization, is this influencing your activities concerning safety biomarker utilization? And so, Tanya, let's go to you first. How is this guidance document, or I might even add some of the IMI activities since you're based in Europe, how has that influenced your utilization or tendency to use safety biomarkers?
Well, actually, I think it's helpful for bringing everyone onto the same page for some definitions and how to navigate through the FDA for development and qualification. But as far as directly influencing activities, I think it more influences our activities through consortia or as far as if you do have a biomarker for a specific study. But I don't think it necessarily guides our strategy at all, or again, helps us understand how we can use them a little bit less conservatively.
Okay. Ernie, from your perspective of...
well, clearly this guidance document is not that old. I think it came out last October, but right. By all means, correct me if I'm wrong. And one of the goals I'd have to say that we hope to achieve today was increase awareness of this document, and the preliminary results, which were just up and came off, still showed that 66% of the audience really isn't familiar with this. So I really think what we want to do is try to make people more familiar that there is a structured process that makes sense and is being endorsed from a regulatory perspective for moving these things forward.
Okay. Liz, I think maybe a little overstatement for this discussion, but I think perhaps what the PSDC has done with the renal biomarkers is in many ways perhaps served as a prototype, or at least framed the thinking of the drafters of this guidance document. But what's your perspective on how this guidance document can enable or encourage people to use safety biomarkers?
I do think that that's true. This process, FDA characterizes this process of growing out of the voluntary exploratory data submission process, or VXDS, and that those renal biomarkers are really the first pilot that was then followed on by a submission from Ilse Hesse with further kidney biomarkers. And I think there are about a dozen or so active qualification submissions right now, currently at FDA in one stage or another. And note that EMA also a couple of years ago released sort of analogous guidance regarding qualification of tools, bioanalytical tools for use in drug development. My great hope is that by releasing this guidance and really formalizing and publicizing this qualification process, that FDA will encourage and that any sponsor that wants to come forward for any drug development tool, but especially in the area of safety biomarkers that we're discussing today, my hope is that we do see more data, more interest, more information coming forward. I think, and the agency is well aware of this, I think what we'll have to be careful about and the risk we run is really the ability to assimilate all that information that comes forward in a very comprehensive fashion. And I think to Tanya's comment, that it doesn't necessarily influence Roche's internal activities, but rather using the mechanism of different consortia to do this does make sense. I think, again, it's been a number of years, like I said, since the kidney biomarkers have gone in, despite consistent efforts from a number of different groups to put this together. It's just really a fairly high bar and a large amount of information that needs to be put together to form a qualification, even when we do organize that qualification into specific context of use. So, we don't go for the whole kitchen sink. We focus on a small area at a time. It nonetheless is a time-consuming process. So, I hope that the agency certainly will have the ability to internally resource and support these qualifications that come through. And as well, I think there may be challenges for them as well as for the different consortia working in this area to make sure that we have mechanisms and frameworks in place to be as cooperative and collaborative as possible in making the ability to put different types of information in context of each other so that, as I said, again, we can assimilate the picture that emerges about the utility of any given safety biomarker in a very comprehensive way.
Okay. Thank you, Liz. But Daryl, you're kind of in the middle of all of this. What's been your experience since the guidance document has issued? Have you seen it influencing perhaps how people are using safety biomarkers?
I can't speak to the use. I can speak to internally there's a high degree of interest. I think that the comment about is FDA going to be willing to resource this enough to move things along are well taken. And I would say that the internal teams are being formed hopefully in a very timely fashion, and then hopefully we'll be able to focus in an efficient sort of way to move the qualification process forward. I guess it's a little disturbing to see that there's not a greater degree of awareness among the participants on the call, and it could be that the FDA's website's not as translucent as it might be. But I certainly agree with you, Jack, that kind of getting the word out. Remember, this is a draft guidance, and so comments are welcome. And that essentially raising community awareness, we hope will also increase awareness of the consortia to encourage participation to move the data gathering and data compilation ahead faster, because that's what will really help us in terms of facilitating getting qualification going forward.
Excellent. Thank you. I'm going to apply the moderator's prerogative here, and I want to jump to question 6. It's been mentioned by a couple of the panelists about the use of histology or histopathology to verify or to validate, if you will, the observation of the biomarker, and with many organizations going to the use of systems biology or systems toxicology or relying more on focused in vitro or ex vivo studies, I think it's important to understand how we can get from those to either in vivo animal or human biomarkers. So question 6, Eric, and I know you have that polling question up. If you wouldn't mind, if you could flip quickly to number 6, and then we'll go back to the polling questions. But the question is, with the rapidly expanding use of systems biology or systems toxicology, how can one leverage the findings in these platforms to define relevant safety biomarkers? So with that, Daryl, maybe I'll go to you first. And I know you're in the middle of this mode of action, mechanism of action activity. Would be eager to hear your thoughts on systems biology or other types of approaches.
Well, as you say, the informatics approaches and coupled with systems biology activities are some of the basis of what we're developing. At the moment, I think it's fair to say that it's in a developmental stage. We're in an early hypothesis testing stage in which we'll be learning with specific projects in specific either disease areas or molecular target areas, the utility in terms of among the oftentimes very large variety of possible markers, the intelligent process to select those that have a high likelihood of utility. I think the comments about then looking to the clinical databases to see as signals to inform that process are right on the mark. And we hope that we can, both with clinical data that is in-house and clinical data that you're producing, help inform the process as well, so that as you said at the beginning, to create that linkage from the clinical to the preclinical, we think will move the process forward much more rapidly.
Thank you. Tanya, what does your group think about observations done in vitro, ex vivo, in silico? How are those teed up or translated into potential safety biomarkers?
Carefully. I think that as more, especially in vitro systems, I think it depends on the question and how you use them for translatability to in vivo. But if designed correctly, I think it's a powerful way to reduce the number of animal qualification studies you may have to use. So, for instance, if it's a simple question of understanding if the biomarker is responding to hepatic enzyme induction, you may use your liver cultures, your single liver cultures for that. If it's a more complicated question, then the 3D cultures that are trying to be developed may be more useful, where you have more cell components there. So I think it's just how the in vitro system is defined that translates to whether it's useful for an in vivo endpoint. And then you obviously have to do a comparison study. I think the difficulty with the systems biology and toxicology that everyone points to is that the bioinformatics, it's very difficult to have that keep up with the amount of data that's generated and interpreting it correctly. And probably part of that comes from that you really need scientists and biologists that are your IT support so that they can better define the program to the question. So I think you know, if you can find that nice mix in your bioinformatics support, that'll be the strongest. And then another, you know, I guess that's probably what I would say as far as using systems tox or biology.
Okay, Liz, what's your view to some of these new methods or platforms being a source of biomarkers?
Yeah, I think, you know, one of the real values that I see potentially coming out of systems biology is, you know, as we gather the information and have the ability to put it together in a meaningful fashion through bioinformatics, the issues that Tanya mentioned, is really the ability to then overlay the human and our tox species signaling networks that we understand to be critical sort of nodes or areas for the allowance of then greater non-clinical to clinical predictivity. And I think that, you know, again, this concept of understanding signaling networks and trying to utilize those for biomarkers, I hope we'll see a shift to thinking more about biosignatures of toxicity. And, you know, again, I think this is going to be a difficult mind shift for us to grasp, rather than really trying to want to put our finger on, you know, our one BLL biomarker of liver injury. You know, we can shift to an understanding of how several things might change in concert to give us different information. Obviously, you know, statistical interpretation and algorithms that need to be developed to actually put those into use for decision making is a fairly large change from where we are today, but I do think that this is possible and where we're headed with more uptake of systems biology in the safety assessment setting.
Okay.
I also think maybe the other utility is when the biomarker does not behave as you anticipate, that you may have information networks to go back to understand what could have confounded how it responded. So like Liz was saying, that there's other integrated pathways and maybe understanding that better.
Okay. So I know we're up against the noon here, but I do have one question, and Eric wants to make one comment. I have a question from a participant that I think is pretty important. But Eric, you want to go ahead and mention the
yeah, I just wanted to mention these two upcoming conferences very quickly. There's a conference organized by IQPC coming up in a couple weeks out in London. A lot of the DSEC members are on the agenda. Ernie Bush is hosting and moderating a panel. It's a great program. Take a look at it. If you've attended today's session, you get 15% off. 15% off, that is the registration. Just make sure you use the code DSEC15 pre-talks. That's DSEC15 pre-talks. And then coming up in May, many of you guys know CHI, our parent company, has their world-renowned Biomarker World Congress, and that's coming up May 2nd to the 4th. And as usual, it's down in Philadelphia, and you could enter the keyword CHA when you register or mention it when you're calling in to telesales, and you will be able to get a discount off that program. And of course, Daryl's boss, along with any DSEC members and others, are on the program. So it should be a great conference. It always is. So those are just two quick commercials before we move on. Go ahead, Jack.
Okay. So I apologize for running over, but I do think this is a critically important question, especially for those who are not in a position to or who have not joined consortia or other collaborative types of activity. Is there a way that people who are not members can gain access to information and feedback from pharma and consortia to guide folks in their own biomarker or methods or technology development? So I think perhaps I could ask Daryl and ask Liz to quickly answer that. Are there ways that folks can get access to these programs and these data? Liz, maybe you first.
Sure. So, we do have a comprehensive communication strategy that, as a consortium and as each of our working groups do develop in terms of usually presentations at scientific conferences are the forum for disseminating, the most up-to-date information about the group's activity. So that's something that, again, we try to list on the Critical Path Institute's website when there will be presentations given by our groups of interest. Publications are obviously another mechanism, but they can be slower mechanisms to get information out. We've also kind of toyed with the idea of doing educational type seminars for the public to learn more about what CSTC is doing on an ongoing basis. So there's the potential to look for that as well.
Daryl, from your perspective, how can folks who are not active participants in these collaborations but have an interest in the data or developing their own activities, how can they get access to this information?
Well, I think Liz covered it pretty well. Certainly the preference would be they join the consortia, and then secondarily, when the data, which is either for the most part or completely into the public domain, and then that's where it is. With regard to beyond that, are there accesses to data within our walls? I think the short answer is not very easily, if at all. But certainly the activities of IMI and CPATH and others are out there in the public. And I suppose if one wants earlier information, the real way to do that is just get more involved with the consortium.
Okay, thank you very much. So we are running a little bit over. I beg the indulgence of our panelists and thank our participants for staying on, even though we did run over. And again, before I close, I would like to thank very much the panelists for taking time from their schedules and to provide feedback to us and respond to these questions. I very much enjoyed it and think it's been very valuable for our membership. So, Eric, with that, back to you.
Thanks, Jack. I want to thank all of our panelists. Thanks so much, which was great. Apologize. We really do try to not go over, so a few minutes late this time, but it was a great dialogue. I want to thank all our panelists and all of our attendees, our members, our guests. We know you're very busy, and 60 minutes, or in this case 65 minutes out of your day is valuable time spent, so we appreciate you spending it with us. I have put a feedback form out there in front of you. There seems to be a few a little technical glitch with SurveyMonkey. At least my view is it looks a little bit funky, but our hope is that you would go in and just provide us with some feedback. It's a quick multiple choice survey. It takes about 3 or 4 minutes at most to complete. Folks on the panel, thanks again. This will mark the end of the call, and we look forward to seeing many of you at SOT next month and then at our next webinar in April. So this will mark the end of the call. Thank you all again. Thanks again, Jack. Great job.
You're welcome. Thanks to all. Bye.
Bye.
Bye-bye. Thank you.